Right. Welcome, everyone.
Dom, just remember to go to presenters mode.
Yeah. Sure. I hope you can all see my slides. Anza, can you please confirm?
Yes, I can see your slide.
Perfect. So welcome, everyone. This is the last webinar series for before the summer break, the 12th session. And my name is Domenica Berardi, and I'm glad to host this webinar together with Anza from University of Johannesburg. And I just would like to remind you to connect with us online through social media. You will find us on Twitter and also on LinkedIn. And here you will see also reported our email contact details. And I'm very glad and honored to introduce today to all of you Prof. Nicholas Rattray and Patricia Kelly from University of Strathclyde, Glasgow in Scotland. They will talk about biomarkers discovery in metabolomics from research to clinical translation. And before starting with this webinar, I also want to remind you about a conference in Osaka in a few weeks, and we will be very happy to see you all there in Japan. And so with this, I'm gonna introduce now Prof. Nicholas Rattray. Is an associate professor in clinical metabolomics within the University of Strathclyde. His group collectively investigate biomolecular changes of aging and frailty through the optics of dysregulated energy metabolism, with the aim of translating metabolomics data into clinically relevant biomarkers panels. Is a Royal Society Industry Fellow and currently sits as the chair of the Scottish Metabolomic Networks. And with this, I will give the words to Prof. Nicholas Rattray.
Thank you, Domenica, and hello everybody. What I'll do, Dom, is I'll start sharing my screen now. So
yeah.
Does that look okay to you now, Dom?
Yeah.
Perfect. Okay. Good morning to everybody in the Americas. Good afternoon to everybody in Europe and most of Africa. And good evening to the rest of the folk in Asia. And good night to everybody in Australia and in New Zealand. And if I've missed anybody else off, thanks ever so much for turning up and listening to myself and Trish Kelly, an RA in my group, and we're going to talk to you today about sort of some of the history of biomarker discovery, and then 3 different projects that we've done in the lab to try to use metabolomics tools to develop biomarkers in clinical health. And as Domenica mentioned, so where my position has previously been within the Metabolomics Society is when the EMN started in 2015, I was one of the first members of the board of the EMN, and it's amazing to see how it's grown and over the last decade it's pretty much taken over the conference. So to back up what Domenica said, if you can get yourself over to Osaka, you'll have an amazing time, and ultimately what you'll do is you'll expand your network, and I think that's the main thing that the EMN has done for me. It's enabled me to expand my network, to have collaborators across the whole world, and we're a cheery bunch in metabolomics, and we kind of all know each other as well, and it'll be a great time over there. As Domenica mentioned, we are at the University of Strathclyde in Glasgow, and a lot of you probably have not heard of the University of Strathclyde. We've had a few famous scientific individuals that have come from our university. No one's so more famous than the guy called John Logie Baird. And what John Logie Baird did, for those of you who don't know, he was the first guy that did the world's first live TV transmission in 1926. So in a building about 400 yards away, or 400 meters away from where I am over there, the first ever TV broadcast in the world, in the history of the world ever, was done. And I think that's pretty cool for a university to have done. So, an overview of the webinar that we're going to do. I will briefly introduce some of the definitions and some of the historical development within biomarker research. I'll talk a little bit about the biomarker development pipeline, and then I'll do a case study. Trish will do 2 case studies, and then we'll have a final word, and then we'll open it up to questions. And just as a quick sort of caveat to this webinar, ultimately, there's an almost infinite palette of biomarkers that can cover flora and fauna. And what I'm trying to say here is that we very much focus on human biomarkers. There are biomarkers for all animals, there are biomarkers for all vegetables and ultimately all growing things. And I think this webinar is very much focusing in the human domain. So
Hi, Prof. Nicholas, sorry to interrupt you. It seems like people are having a bit of an audio challenge. Is there a way to move closer to your mic?
I can move closer to my mic if needs be. Hold up 2 secs.
Yes, please.
Can you hear me better now?
I can hear you better now.
Can you hear me better now? It's okay. I've just moved my microphone underneath me. So that's fine. Any other issues, just let me know and I'll try and make adjustments, okay?
Awesome.
Okay, perfect. Thank you. So where was I? Biomarkers, it's quite a broad terminology, and they're so important. The World Health Organization, the United Nations, and the International Labour Organization have a unified definition of a biomarker as any substance, structure, or process that could be measured in the human body or its products that can influence or predict the incidence and outcome of disease. It's quite a mouthful, but ultimately what a biomarker really does is it can indicate something. It can tell that an event has happened, that a change has occurred in a biological system. And there's lots of different biomarker classes, and this is not an exhaustive list. There are several other different subgenre of biomarker classes, but the main primary ones that are considered within clinical research are diagnostic biomarkers that are used to detect or confirm the presence of a disease or a condition. Monitoring biomarkers that are usually measured serially for assessing the status of a medical condition or the use of a medical product or environmental stimulus. Prognostic biomarker to identify the likelihood of a clinical event or a disease recurrence. Susceptibility, I suppose a lot of these biomarkers, the devil's in the details in the name, which means you're more susceptible, for a susceptibility biomarker, you're more susceptible to developing a disease or medical condition. Predictive enables an individual to identify if you're more or likely unfavourable events going to happen on an exposure event or upon treatment of a medical intervention. A response biomarker or a pharmacodynamic biomarker is used to show that a biological response has occurred to an individual that's been exposed to a medical product. And a safety biomarker is measured before or after an exposure or a medical product or an environmental agency. Like I said, there's a lot more of these different biomarker classes, but most of them are siloed within these different classifications. Now, the thing that we're probably more interested in are the actual types of molecular biomarkers that are out there. And these cover, extensively, these cover the central dogma of molecular biology. We'll talk a little bit more about that going forward. They range from RNA and DNA-based biomarkers. Most of us have probably heard of the BRCA1 and BRCA2 genes being expression being linked to breast cancer and ovarian cancer risk. Also with proteins, things like prostate-specific antigen or PSA being linked to prostate cancer detection that was developed in the 1990s. And then almost since antiquity, and we'll talk a little bit about this shortly, glucose levels have been known to be an indicator, elevated glucose levels have been known to be an indicator for diabetes. And it's been since the 1960s that AIMS developed the first sort of quantitative glucose blood test kit that then allowed individuals to really be able to accurately measure what the levels were, and then to have dietary intervention to try to circumvent the levels that they've got within their bloodstream. Other types of biomarkers that are out there are things like cellular biomarkers. If you think about your immune system and immune function with patients that suffer from HIV, they very much count their CD4 cell count. When that goes really, really low, that can be an indicator of a really poor low immune function. And other biomarkers can cover the physiological realm, which include blood pressure, heart rate, and imaging biomarkers. Now, imaging biomarkers are quite interesting because they've been around ever since humans and hominids have had eyes. And it's only really since we've developed more imaging techniques such as MRI and CT scans that we've been able to not just observe and document a malady, or not just observe and document a clinical change that's happened with an individual, but we've now got tools like MRI and CT that can allow us to track those internally with individuals as well. And the real importance behind biomarkers, and the reason why I suppose metabolomics is really, really important within this area, there's two reasons. There's the justification that 70% of all medical diagnoses are dependent on clinical lab diagnostics. Now, the vast majority of us here have probably had some form of blood test taken, and in the Western world, 70% of all disease that is diagnosed uses some form of ELISA, some form of mass spectrometry assay, or some form of spectroscopic assay to be able to determine if a person's got a disease or not. Last year in the US, there was over 7 billion of these clinical biochemistry tests that were performed. Now, I couldn't really work out what the stats were globally for this, but I could probably estimate you're looking at sort of 50 billion to 70 billion clinical biochemistry tests are performed every year, which is a ridiculous number. That is a lot of tests that are performed. And globally, this costs nearly $250 billion. And for those of you that are interested in the finances of it, that's got a compound annual growth rate of about 14%. So that means every year that number goes up by 14%. And then the new number goes up by 14% the next year. And then the new number goes up by 14% the year after. Projected by about 15 to 20 years' time, that'll be over $1 trillion per year. And a lot of my research has been mentioned at the start by Dom is related to aging and frailty. And through those optics, age-related disease burden accounts for 51.3% of total disease burden. And the reason I'm saying that is because biomarkers for aging and frailty, if you think about the fact that half of disease has a confounding factor related to age, the more we understand the processes and the more we can develop biomarkers in that area, we can potentially then reflect on that disease burden as well. And the other important factor about why metabolomics and biomarker development, biomarker discovery sort of go hand in hand, I suppose, is because, what do our technologies do? Well, our technologies allow us to tell the difference between classes. And that's ultimately what biomarkers do as well. Our mass spec, our NMRs, when we run them and we classify our samples and we do our PCAs and all of our other statistics, we're separating our samples into groups. And ultimately, that's what biomarkers do as well. And that's why the technologies and tools that we have are so sort of married and so well aligned for this sort of research and this sort of work. So going on to a little bit now about the history of biomarkers, and I truly believe that our ability to use our eyes and our brains and to assess what's in front of us and to make decisions about how stuff has changed has been a capability that almost pre-human existence has probably been available from different animals. And as we've developed as hominids and then into humans, we've been able to measure different things that have changed within us and our families and our groups. And that's gone on. It's ultimately, I think that area of almost very crude diagnostic ability or categorization ability, I think supersedes our ability to document it. It supersedes our ability even hundreds of thousands of years ago when there was cave drawings. I think we were probably able to work out if we had blood in our urine, there was probably something that was wrong with us internally inside us, and we just didn't really have the tools to be able to express it. And those tools started to be developed or have been categorized and started to be developed round about 6,000 years ago, where certainly the Egyptians and scientists in China noticed that ultimately they played around with urine quite a lot, and they managed to determine that via the smell and the colour, that might be an indication of disease. If ants were attracted to the urine, it might be an indication that somebody could be diabetic as well. So even back then, 6,000 years ago, where the technological advancement that those civilizations had were completely dwarfed by what we've got now, they were still individuals that were clever enough, had a brain, had observational skills like we've all got, could still make really critical decisions and really important diagnostic decisions that still hold true to this day 6,000 years later. Imagine if you discovered a biomarker now that in 6,000 years' time, they're still using that. In 6,000 years' time, an academic is talking about and is still sees that as being worthy. If we fast forward now to 500 years BC, scientists like our philosophers and scientists like Hippocrates and Galen were very interested in the idea of humorism. What humorism did was, again, it categorized the fluids within our bodies, and it looked at things like black bile, yellow bile, red bile, and blue bile, and looked at the fluids within our bodies and managed to try to use those to justify diseases and health conditions. again, the methods that they had were really, really crude. And often the method that they had, again, was their eyes and their ability to dissect animals and ultimately to dissect people to be able to make the observations that we've got. Thankfully, we've got ethics that means that going forward, we don't necessarily have to do as much of that anymore. And we've got tools that can allow us to visualize or internalize some of those diagnostic procedures. But I couldn't really take sort of the history of biomarker detection and discovery without sort of adding a Scottish flavour to it. So I don't think there's going to be many of the 99 people on this webinar that will know what that dwelling is called. So it's called a crannog, and what a crannog is, it's a house that has been built on a loch or a lochan or a lake, ostensibly within Scotland, Ireland and Wales, and they date back from Neolithic times, so about 4000 BC, all the way up to about 500 AD. And it's where people lived, it's where they kept their livestock, it's where they kept their grain. And what this group, what Brown et al. Have done is they've looked at some of the archaeological digs at some of the sites for crannogs, and basically you can see where they're located very much, if I just bring my pointer on here, very much around Scotland, around the north of Northern Ireland, around Northern Ireland and north of Ireland, and Wales here. And what they did was, it was the first evidence they could find in the literature of lipidomics being used within Scotland and Ireland. So over 2,000 years ago, they took samples from over 2,000 years ago, and they looked at bile acid distribution within these samples. And from this, they managed to, from the remains of humans, they managed to determine diet, animal husbandry, and some of the reasons why those individuals might have died and might have had healthcare issues. So you can see even from archaeological dig sites, you can still use our tools in biomarker discovery and biomarker history to ultimately uncover some quite interesting artifacts, all over, not just in Scotland, but all over the world. So moving forward from 6,000 years ago now to more the medieval times of about 500 years ago, now we're in the realms of where individuals had ability to, I suppose, had ability to observe and document far better. Our ability for literature and art had become on orders of magnitude better, and individuals like Vesalius, who was a leader in human anatomy, laid the foundations of ultimately what our body was made up of. You know, they managed to, in exquisite detail, be able to document where our organs were and potentially hypothesize how different systems within the body, say the blood system, the lymph system, were linked to different organs. And round about the same time, a couple of decades later, William Harvey very accurately managed to map out the circulatory system within our body. And without knowing that information, how would we know anything about blood-based biomarkers? How would we know anything about the fact that molecules can traverse our whole body and travel through the whole circulatory systems, and ultimately leads on to that sort of idea of blood-based biomarkers? And alongside this, we've got Leeuwenhoek and Hoek, who ultimately, both of them were synonymous with developments within the microscope, which allowed us then to look exquisitely at cellular detail and develop cellular biomarkers from this. And again, without all of these developments, our knowledge and our ability to categorize stuff, our ability to categorize information, it wouldn't be to where it is today with our modern technologies. And speaking of which, you know, moving forward into the more modern times, you know, round about 124 years ago, Landsteiner developed this idea of blood typing. So again, being able to categorize blood into A, B, AB, and O positive and negative blood types, which was critical in being able to develop our understanding of how blood works and potential transfusion. Fast forward 20 years, Best and Banting discovered insulin, again critical for that glucose monitoring, for that ability to deal with diabetes. But it was at that point where everything changed, because just after the Second World War, and a lot of the technological advancements that we actually got from the development of the atomic bomb, particularly GCMS and particularly mass spectrometry technology, is where our NMR development, our GCMS and our LCMS really began to take traction, really began to be developed and then implemented towards the first use of metabolomics and the first terming of the coin by Oliver in 1998. Now, I could give a 2 or 3 hour lecture just on the history of biomarkers, but obviously for the sake of time, most of you will probably be aware that Gary Sustak, a lifetime fellow of the Metabolomics Society, has just released a free book. If you do want to clue yourself up a little bit more about biomarker history and all things activity metabolomics, click on there and you'll be able to get yourself a copy of it. But if we now zoom further in on that time point of 1998, as I move on a little bit, that time point of 1998, you can see this is when our area began to open up, and our ability to be able to utilise different technology to detect different biomarkers opened up far more. So in 1999, the first NMR or metabonomics experiment was first documented. GC was first used in 2020 to do an untargeted experiment, and a lot of these first experiments that were done, particularly for GC, were focused more on plant-based metabolomics or microbial-based metabolomics. Then the first International Metabolomics Conference took place in Wageningen. And then one of the next breakthroughs was being able to apply high-resolution mass spec-based metabolomics. And I think where the high-resolution part becomes really, really important is because previously, before this, we only really had the separation out in the chromatographic domain. But being able to develop instruments in the mass spectral domain that had a resolution at that point of about 5,000. Now we have instruments that have resolution of 1 million and 1.5 million. Back then it was only maybe a resolution of about 5,000. We could still add more data into our models. We could still identify more features within our models. And as we move through, it was 20 years ago, as you can see, 20 years ago, the Metabolomics Society was constituted. A year after that, Roy Goodacre started the Metabolomics Journal. And from there, everything just accelerated. We got the Orbitrap, the Metabolomics Standards Initiative, iMobility was used, and then this term that everybody's beginning to use within the data sets is AI was first used in metabolomics. And machine learning has actually been used for far longer within metabolomics, but AI was first used. And moving on a little bit further, so as I've kind of alluded to, it's only really by the implementation of not just metabolomics, but proteomics, transcriptomics, and genomics that we can actually understand the biomarkers that we develop. It's one thing understanding, our dysregulated chemistry that happens in that one domain, be it the metabolome, but we really need to understand all of this chemistry to have a full understanding of what's actually going on within a disease. And if you want to get your results into the really big journals, you really have to begin to track your results into these different domains. And the way in which technology has been used and the way in which technology is developed in these different domains actually has sort of two main different concepts that kind of transverse each other. And allow me to explain. When we begin to look at the genome and when we begin to look at the transcriptome for biomarkers, it's really complicated. There's 3.5 billion base pairs within your genome. But when you actually begin to look at the chemistry of your genome, you've only really got 4 bits of chemistry. You've got your A's, your C's, your G's, and your T's. If you look at the transcriptome, you've got thymine and uracil, they're swapped over. Again, you've only really got 4 bits of chemistry that you've got to detect to be able to compare and to be able to develop the biomarker and ultimately that bit of chemistry that's changed. And this is why gene sequencing technologies are, I'm not going to say they're far further ahead than metabolomics technologies, but that simplicity in chemistry means they can standardize their methodology way better and way more robustly. When it comes down to the proteome level, again, when you think about it, and you think about all proteins, and you think about the protein coding region in your genome, which is about 1%, and your exome, ultimately though, it's only really 20, 21, 22 if you take selenocysteine and selenovaline into the equation of the protein coding region. There's still only 21 amino acids that you've got to measure. And if you look at the protein coding region in humans, there's about 22,000 proteins. If you look at the post-translational modifications, there's about 800,000, maybe about 1.2, 1.3 million now, I think, in total. If you look at the conformome of proteins, of the shape that they take, I mean, that's almost infinite, but that's again for another talk. But again, I always go back to the fact you're still only measuring those 21 amino acids and the peptides that they form together. So what do we have when we think about our metabolome? As I've gone down there and talked about, I'm talking about diversity in chemistry. And our diversity in chemistry that we have to measure when we're measuring or trying to develop biomarkers is inherently complicated. If you look at this is basically a picture of the pathway map behind Trisha behind me. This is just under the canonical pathways of just under 400 metabolites within the human metabolome. And I'm pretty sure most of us have been in the human metabolome database at one point. They're way north of a quarter of a million different metabolites that have been quantified or predicted to be present within human bodily fluid. So I suppose when you're looking for biomarkers within humans, the great thing about metabolomics is there's over 250,000 potential different biomarkers that can form a biomarker panel. The disadvantage of metabolomics is the fact that there's 250,000 different metabolites that we've got to understand all of this complex stuff. And this is where AI, machine learning, these prediction tools that we've got become really, really important, because the stuff that we do is difficult. And, you know, it's really, really, you know, the fact that we've got this really big imbalance in the chemistry that we have to measure means that we have to refine our technologies and really, really refine the methods in which we measure metabolites. And the way in which we measure across the central dogma, again, these sequencing arrays, these have been really, really standardized by the community. For proteomics, the methodology primarily used for shotgun proteomics is a C18 PipMap column, usually over, sometimes you do ballistic gradient, usually over an hour to 90 minutes, sometimes short. But again, that chemistry is really, really quite refined. And I've got one proteomics method that I use in my lab that kind of covers quite a lot of the ground. For metabolomics, we've actually got 8 different LC methods that we'll use for metabolomics in pause and egg mode. We've got a C18 method and 3 Hillick methods that are adjusted according to pH. So again, that's the devil in our detail. It's almost the Achilles heel that we have within our area, but it also leads for lots of different method development parts. So that's ultimately a positive side to it. It's a positive side to it as well. So applying all of this to the biomarker development pipeline, there's 4 main areas of that pipeline in which we can really apply it. And where we operate ostensibly is within this discovery section, where you're looking for things that change. You're using your own target methodology to maybe look at 50, 100 different samples. It's usually quite difficult to do a power calculation to work out exactly how many samples we do. There are AI tools in order to try and calculate that, but often we say 30 or 40 samples per class and have at it and see if you can get something that comes out of that experiment that you've done. You basically try to identify a smaller panel of maybe 50, 20, 10 different metabolites that you can then move on to the analytical validation stage. And this is where you need to become, as Roy would say, a little bit more belt and braces with the work that you do. You need to contextualize those biomarkers, i.e., do they relate to the biological question that you're talking about? Is there some precedence within the literature? You then also need to potentially develop a more targeted assay. You know, can we target it? Can we look at precision, sensitivity? Have we actually identified the thing properly? Have we actually done it right? And this, again, this part can take, you know, in the realms of months to be able to do properly, depending on the number of samples that you've got. I'll use it as an example. The case study that I'm about to talk about frailty, and we were lucky enough to get that paper published in Nature Comms. It sat for 2 years in Nature Medicine under review, simply because we got to this next stage of clinical revalidation, i.e., have you done it in another clinically relevant data set? And ultimately, one didn't exist across the world. There wasn't the same data set we could possibly cross-validate across it. And I think, again, going back to, if you want to get your work published in the highest journals and move through and ultimately get your biomarker to the clinic, you have to have line of sight of doing that discovery piece, identifying your biomarkers, and then ultimately rolling it out into a far larger prospective and retrospective clinical trial. And we might be talking thousands or tens of thousands of individuals that you need to collect samples from. You then need to evaluate the clinical utility of that as it goes forward. And again, that's not a trivial task. That's something that will take millions of dollars or pounds worth of funding. It'll take close collaboration with clinical colleagues to be able to do it. But if you do that as well, and your panel of biomarkers still holds out, then you're in a very interesting space, because then if you can go through, in America would be FDA review and qualification, we'd probably go through NICE and through the NIHR for qualification here in the UK. And if you can still manage to do that over multiple centres and prove that your biomarkers can be and have some form of diagnostic utility, then you've got the potential to not only potentially save millions of lives, but ultimately roll out a piece of work that could ultimately, define your career and define the work that you've done. So it's a huge process. Often your biomarkers will fall down in one of these 2 or one of these three main areas, but the reward at the end is you could actually make something that can make a difference to a lot of individuals. So that's the end of the first part of the talk. The next part of the talk is going to be given by myself, and it's going to be looking at frailty as a case study of discovery-based biomarker development, and looking at aging and looking at, so I suppose to define what frailty is a syndrome associated with aging where an individual suffers from a minor stressor event where somebody who isn't frail would recover from. Something that could be a bacterial infection, a fall, even something like a loss of money or a death of a spouse or a partner can cause somebody to have this major stressor event. When you're frail, you don't necessarily recover from it, and ultimately it leads to way poorer clinical outcomes, and it leads to the person dying sooner than they would do if they weren't frail. And in order to understand frailty, in order to use the tools that we've got and understand how the chemistry changes, this paper by Lopez-Otin came out just over 10 years ago that identified the main hallmarks of frailty, and main hallmarks of aging. And what you can really see is they're hung over the central dogma of molecular biology. These can all be hung over that. And these are some different aging-associated main cellular processes that can be mapped out. And this study that actually I performed or started to perform in Roy's lab, as I mentioned, it took about 5-6 years to do in total, just the discovery part of it. So I was a postdoc at the University of Manchester. I then went over to join Caroline Johnson's lab, actually where Domenica is now, over at Yale. I postdoc for 2 years, and then I started my own group over here at Strathclyde for 2 years. By the time we actually got this whole project out, so it did take a while. And the idea behind it is to understand the metabolism that's changed within individuals that are frail within community. So, and what we did was we worked very closely with the English Longitudinal Study of Aging, or ELSA as it's known. Within ELSA, they've collected blood samples from about 12,000 individuals across the northwest of England over an extended period of time, about 15 years. And what we did was we took blood samples and we did our metabolomics magic, and we'll talk about that in a second. But we also developed this thing called the Rockwood Frailty Index, which is called an accumulation of deficits model, where a clinician or a research nurse has taken an individual, they've taken a blood sample that we did metabolomics on, but they've also taken 50 or 60 anthropometric measurements of that individual. How long does it take them to put on a jacket? How long does it take them to walk to the door? What's their psychology like? What's their diet like? And if you take all these measurements and you put them in a population level, in this case it was 1,200 people to start with, you get a thing called a right skewed or a positive skewed distribution, where the individuals over here, they're elderly, so they're older, they're over 65, they're fit, they're fitter, they're healthier, they're still cycling, they're still playing with their grandkids, they're still in a good healthy place. The individuals over here are not. They could be bedbound, they could be homebound, and they need a lot of support from their healthcare provider, from their family. And the cynic in me say they cost society a lot of money. A frail individual in the UK that goes into primary care, so that's like a GP surgery, costs 4 times more than a healthy age-matched individual. If you end up in emergency care, it can be something like 10 times more. So the idea then was to see, well, what metabolic differences are different between those at this end and those at this end over here. So we did this work, and what we found was, this was again, Roy was the senior PI in the work. What we found was that there was dysregulation in 3 main areas. Vitamin E was dietary related. We didn't really investigate that much further. But the monosaccharide pathway and carnitine shuttle pathway are very closely related to the ways in which our mitochondria generate energy. And it kind of makes sense that those individuals that are frail have a lesser energy output. Basically, there's been a lot more dysregulation in the way that they make energy. And associated with that, we've got the kynurenine pathway, which is related to the way that we make and recycle NAD. It's also related to the way in which NAD then relates to how our inflammatory system operates, because kynurenine is also related to the way in which prostaglandins are expressed and then ultimately start that whole inflammatory cascade. And when you get older in your life, your inflammatory response ultimately has more of a memory, and it tends to be a little bit more excited, in inverted commas, and then this concept called inflammaging kicks in, and some individuals suffer from that more than others. And it's been known that individuals that are frail have a more sensitive inflammatory response. So we again saw elements of that within this metabolomics experiment that we did. So we got a panel of 25 individual metabolites. We then cross-validated this in our second cohort to satisfy the reviewers of the paper, of the journal, and also to validate our analytical technologies. And ultimately, we got a panel of 13 biomarkers that with a ROC curve of just about 0.8 could be used to predict if an individual was frail. And nobody had ever done this before, and ultimately we were really happy because nobody had ever done this before, and I think at its time it was one of the biggest metabolomics projects that had ever been performed worldwide. And when we got this data, when I was setting up my group over here at Strathclyde, I had to think about what I was actually going to do with this data and how could I make use of it and how could I utilise it. So going on to the second sort of aspect of the frailty work that we do in the lab, I tried to then think about how we could then utilise this in the surgical setting. And I'm very keen to look at gut health as well, and that links on nicely to the work that Trisha's going to talk about in a couple of minutes. And if you're over 65, if you're clinically classed as being frail, and if you have to have emergency laparoscopic stomach surgery, 15 to 18% of those patients don't make it past 90 days. So that's not because of the disease they've got, it's because that insult that they've had, and because they're frail, ultimately has led them to die off the back of it. And obviously, if we can develop a biomarker that can predict those individuals that can survive surgery or not before they have it, that would be super valuable, and that could ultimately save lives at the end of it. So working very closely with Jean, who has made a superhuman effort with this clinical trial at Glasgow. We've also got a secondary trial going on at the University of Sheffield that's dealing with more, instead of emergency surgery, it's dealing with elective surgery. The idea behind it is to measure frailty, take blood samples before surgery, directly after surgery, and at 3 time points up to 90 days, and then do all of the omics stuff that we can do on those samples. And then can we model it? How can we model it? How can we potentially develop those predictive biomarkers at the start of it? And where we are with this work, I'm only going to talk a little bit about this because the results are still coming in and can't talk too much about it. Working very closely with Prof. Dave Broadhurst at Edith Cowan. Dave's been great in helping us and also performing our clinical, our data and analysis strategy. And what we've found within this data is we can begin to, I'm not going to talk so much about this plot, but we're beginning to be able to substratify frailty pre and post-op, which is super duper interesting. Again, it's something nobody's ever done and used our technologies to do before, but we think there's some really interesting biomarkers that might allow us to substratify frailty. We've not looked at that first sample yet before surgery, but that's the next part that we're going to do within this. And it's just to give you an idea of that whole, how our technology can potentially be used in the realm of biogerontology and frailty. So that's the end of case study 1. I'm going to swap seats now, Tricia, and you can take over in the hot seat, and we can now talk about the case study 2 in celiac disease.
So hello everyone, hopefully you can all hear me. As Nick was alluding to, in our lab we do a lot of work using metabolomics as a tool to investigate different diseases, and a lot of the work that I do as part of my research is investigating that in terms of gastrointestinal disease. So as a first case study, we will look at celiac disease and how we can develop a pipeline to look at exploratory biomarkers for the disease. So as a start, we'll look at how celiac disease occurs and its pathology. So celiac disease is a gastrointestinal disorder of the small intestine, and it occurs in genetically predisposed individuals. It has a global prevalence of around 1%, so it is quite prevalent in the world. It's a common disease, and it's quite well characterised in terms of its pathology relative to a lot of other diseases. So we know what causes it, and we know relatively well what happens in terms of the response. So when gluten is digested in genetically predisposed individuals, gluten is deamidated by tissue transglutamidase, and in populations with celiac disease, there is alterations in this process whereby the gluten is not fully processed, and this therefore translocates through the small intestinal epithelium, and this elicits an inappropriate adaptive immune response. So this immune response is directed towards the gluten cells, the gluten proteins, which are gliadin proteins. And we can look at these, this pathology and metabolites that are implicated in this process. So although the disease is quite well characterized, there are a lot of problems in terms of its diagnosis. So it's relatively misdiagnosed, and although 1% of the population is known to have celiac disease, it is very misdiagnosed and underrepresented. And this can cause a lot of problems, as if an early diagnosis isn't occurred, or if disease is not diagnosed, this can lead to a worse prognosis and also worse symptoms. So in a study that was published a couple of years ago from our group looking at alterations in the microbiome of patients with celiac disease, it was found that celiac disease patients had a different microbiome compared to healthy controls. And this contributed to an increasing body of knowledge that now implicates the microbiome in the pathogenesis of celiac disease. So where metabolomics comes in is that we can see that, as we know, the gut microbiome produces a diverse range of metabolites that are implicated in different biochemical processes throughout the body, and metabolite alterations are implicated in different stages of celiac disease pathology. So particularly epithelial translocation, gliadin processing, and then the inflammatory immune response. So the main aim of this study was to characterise the fecal metabolome of children with celiac disease to facilitate disease understanding and see if we can try and develop biomarkers to look at disease diagnosis and try and aim towards that earlier diagnosis as well as disease monitoring. So we used the same patient cohort that was used in this microbiome study. And we had 4 different patient cohorts here. So we had healthy controls, patients with untreated celiac disease, so samples were taken here at their time of disease diagnosis. We had patients with treated celiac disease, so these were patients with pre-established disease and already following a gluten-free diet. And we also had a cohort of unaffected siblings of the treated celiac disease patients. So we then formed metabolite extraction, and this was done using an optimized methods. So we tried to standardize a method for fecal extraction, and we published this recently. And just as a quick summary, we used 50 milligrams of freeze-dried fecal samples in a methanol extraction. And we then acquired the data. We used both untargeted and targeted metabolomics approaches here to try and get an idea of what's going on with the metabolites in the disease. So we had 171 study participants in total, aged between 2 and 15 years. And we also had measurements such as tissue transglutaminase. So this is used in the current diagnosis and is in the current diagnosis guidelines to have 4 times fold TTG levels for a positive diagnosis, as well as the PEDS-QLGS score. So this is a quality of life inventory score. And another advantage of this study was that we used both cross-sectional and prospective study cohorts. So the patients that were samples were taken at the time of diagnosis were also followed up at 6 and 12 months of following a gluten-free diet and recommended to follow a gluten-free diet. So first of all, looking at the results from investigating the disease mechanism. So what we did here was look at the comparison between celiac disease patients and healthy controls. And these were celiac disease patients irrespective of their treatment status. So celiac disease patients that were following a gluten-free diet and not following a gluten-free diet. The reason why this is important is currently diagnostic guidelines state that gluten has to be consumed prior to diagnosis to confirm that it is celiac disease. And this gluten challenge can be quite difficult, especially for children. A lot of parents don't want to re-give their children gluten if they suspect that it's celiac disease. So the comparison between celiac disease patients, whether or not they've had gluten, and healthy controls, therefore eliminates the requirement for a gluten challenge prior to diagnosis. So looking at the separation between the two groups, there is separation between the overall fecal metabolome, as we can see from the OPLSDA on the top left-hand side there. It can be noted here that the model performance characteristics aren't great for the overall fecal metabolome. So our R-squared value is a measure of the correlation index, and Q-squared value is the predictive ability of the model. So while the overall fecal metabolome isn't super differential between the two groups, what we can then do is dive into the data a little bit more and have a look at specific metabolites that are altered between celiac disease patients and healthy controls. So when we carried out this univariate analysis, we found that 119 metabolites were significantly altered between the two groups. 94 of these were significantly decreased and 25 were significantly increased in celiac disease patients. And when we do enrichment analysis, we can see that oxidation of branched chain fatty acids and very long chain fatty acids, as well as alanine metabolism, were our top highlighted pathways to be implicated between these two groups. Importantly, we highlight 24 celiac disease specific metabolites. And these were done using the criteria of having a VIP score from the OPLSDA model of more than 2, as well as being significant in p-value and fold change of greater than 2. And here we just highlight the top 5 of these differential metabolites. So now looking at the effect of a gluten-free diet. while we can see the model characteristics are a lot better for this model, so our R-squared value is 0.7, so it's a greater separation between these two. And this is reflective of the dietary nature of disease. So gluten, your microbiome is a reflection of what you consume in your diet, and your metabolites that you produce indicate your microbiome profile. And when we look at the volcano plot for this, we see 57 differential metabolites, and 40 of which were decreased in the celiac patients. What's interesting to note here is that we carried out this analysis investigating untreated celiac disease compared to treated celiac disease, but also compared to healthy controls. And there were 12 metabolites which were equally altered between untreated and healthy controls, and 45 were specific to untreated celiac disease. And again, we can highlight here some of the pathways that are specifically altered upon a gluten-free diet, and these include alanine metabolism, glutathione metabolism, and pyrimidine metabolism. And we also highlight the top 5 metabolites that were altered upon a gluten-free diet. So looking at this data as a whole, so this is the cross-sectional data whereby we just took all the groups before following them up, and we can look at this in our PC-CVA, and we can see drift from healthy controls to treated celiac disease. Now, at a glance, what you might think, you might expect untreated celiac disease to be further apart to the healthy controls than treated celiac disease. But again, this is most likely a reflection of the dietary nature of disease and how the fecal samples are a reflection of dietary intervention. So treated celiac disease this is the treatment with a gluten-free diet. And here we also used gluten aminogenic peptide. So this is a biomarker for gluten consumption. So in our analysis of treated celiac disease, we only used patients that were confirmed to be following a gluten-free diet. So in this cohort, we had patients that were confirmed on a gluten-free diet, and when you haven't consumed gluten, this will significantly alter your overall dietary profile, which is reflective of these results here. So now looking at our longitudinal cohort, we assessed our patients at 6 and 12 months on a gluten-free diet. What was interesting to note here was that there was a more significant alteration at 6 months compared to 12 months on a gluten-free diet. And while there are metabolites that change longitudinally along with time, we also have the general profile is that there's a more significant difference at 6 months. This likely indicates that either adherence is greater at 6 months because patients are newer onto this diet, or some metabolites might change at the start and then they stabilize after 6 months on the gluten-free diet. This also correlates to TTG and inventory of life scores. So next we looked at our cohort using targeted metabolomics, and we used a method of primary metabolites, and here I have just highlighted the results from the amino acids. So looking at celiac disease versus healthy controls, the key highlights here was that there was decreases in histidine, cholic acid, threonine, and xanthine, and upon treatment with a gluten-free diet, we had increases in aspartic acid, malic acid, serine, and ornithine. So we here see 2 distinct metabolic profiles from our celiac disease patients in comparison to a gluten-free diet, and this distinction is greater than the effect of the disease pathology originally. And from the prospective cohort, we see decreases in threonine, cysteine, and histidine, all of which have important roles in intestinal homeostasis. And these increase upon following a gluten-free diet. So from our celiac study, we see that individual metabolite differences were seen between celiac disease patients and healthy controls. And what's important here is that we identify a panel of 24 celiac disease specific metabolites. And again, this is important as these were celiac disease patients irrespective of their treatment status, so thereby eliminating the requirement for gluten to be taken for these metabolites to be observed. The profound alteration in the fecal metabolome wasn't observed. However, this also matches the study that was mentioned before, looking at microbiome. There was also not a profound alteration in the microbiome between the 2 groups. However, there were distinct specific bacteria that could be used as biomarkers of disease. So this nicely aligns with the microbiome study. However, what we do see is a more significant difference upon treatment with a gluten-free diet. we see 45 metabolites that were altered between the two groups, and this is supported by our longitudinal data set. So we can kind of conclude that we have three distinct metabolic profiles, one of healthy individuals, one of celiac disease patients, and one of celiac disease patients on a gluten-free diet. And what we can know is that on a gluten-free diet, while the metabolism profile is altered, it is not restored back to normal levels. So next, we looked at inflammatory bowel disease. So we can use this pipeline to investigate celiac disease, and next we're looking at IBD. So IBD is also a chronic intestinal condition. Two main diseases are characterized, including Crohn's disease and ulcerative colitis. It follows a similar intestinal pathology. However, while we know the etiology of celiac disease, we don't know what causes IBD. So the main characteristics are degradation of the epithelial barrier, which is then results in overactivation of the immune system towards these translocated bacteria. So there are lots of available treatments that are used for IBD currently. However, they're associated with side effects, loss of response, or primary non-response. So dietary management is an alternative to pharmacological approaches, which are desirable to patients and also clinicians. So exclusive enteral nutrition is a nutritionally complete liquid diet, which is shown to induce remission in 80% of pediatric Crohn's disease patients. So this is used as a first-line treatment for IBD currently for children. And it works super well. It can be given either orally or nasogastrically, and it's used as first-line treatment. But it does provide difficulties because it can only be used for 8 to 12 weeks, so it has limitations in terms of prolonged use, and it's a short-term treatment. So this is where CD Treat was developed. So in Glasgow, the aim of the study was to look at replicating EEN but using a solid food diet. And this was done by excluding certain dietary components, so for example gluten and lactose, and matching of others, for example different micronutrients and fibre levels. And primary results show that this also induces 70% remission in pediatric Crohn's disease patients. However, as I mentioned a wee second ago, this enteral nutrition can only be used for up to 8 weeks, and the same goes for CD Treat. So you use CD Treat or EEN as an induction therapy, and you see healing of the gastrointestinal system, you see mucosal healing, your inflammation levels go down, and after this 8 weeks, food is reintroduced. And once food is reintroduced, inflammation returns. So there's challenges in this phase of food reintroduction. So this is where the IPEN study comes in. So the IPEN study is the intensive post-EEN study, and it's a randomized trial to evaluate CD Treat as a reintroduction regime. So what we have here is patients that have successfully responded to a liquid diet. CD Treat was then used post-EEN instead of an unrestricted diet to assess whether the gut inflammation levels are improved compared to an unrestricted diet. So children and young adults with Crohn's disease who have successfully responded to EEN were recruited and randomized into one of our two groups. So this is pilot results. So this is 6 patients, it's initial 6 patients in each group. The current larger study is ongoing. This is just a pilot study to assess the initial results. The aim of the study was to evaluate the difference in gut inflammation and metabolite levels between the groups after 3 weeks of intervention. So looking at the results, what we can see, we also looked at a cytokine analysis. So what we can see is, so time point A is when patients had just finished EEN and hadn't started either CD Treat diet or their unrestricted diet, and time point B is 3 days after either intervention. So what we can see is that as short as 3 days after either intervention, so after food reintroduction, the inflammatory cytokine levels increased again, and this was a trend that was observed for all of the cytokines. But here we're going to focus on the metabolomics data. So what we can see as a general overview, so looking at the PCA for all the 3 groups, we can see that healthy controls have a distinct profile compared to the unrestricted groups and the CD treat groups. But the CD treat group has a closer metabolite profile to the healthy controls comparison to the unrestricted groups. And we have some metabolites that are shown to differ between these groups as well, having again showing a closer profile to healthy controls in the CD treat group compared to the unrestricted group. So looking specifically at unrestricted versus the CD treat diet, we again see clear differentiation between our two groups. Our model characteristics are looking really good for this one. So we have an R squared and Q squared of 0.8, and 27 metabolites were shown to be specifically altered, with 20 increased and 7 decreased. The pathways which were significantly increased or significantly enriched include phenylalanine metabolism and thyroid hormone synthesis. And we can see our panel of metabolites that were increased. Again, I've just shown the top 5 here, all of which were shown an increased level in the CD treat group compared to the unrestricted diet group. So next, looking at the longitudinal data. So this patient cohort was followed for up to 30 days. We have taken samples and done metabolomics analysis up to day 21. And looking at the unrestricted diet, so this is the comparison of the end of the liquid diet compared to day 3. So this is the first measurement after having consumed food. And we can see that there's a significant difference in the metabolome profile. Now we know that food reintroduction causes inflammation and is shown to be associated with gut inflammation. So what we want to look at is how this unrestricted diet compares to the CD treat diet. So this is a comparison of the CD treat to the unrestricted diet. And what we see here is a separation of these two groups. And 4 metabolites were shown to be different between these, all of them at an increased level compared in the CD treat group compared to the unrestricted group. So we also wanted to look at the endpoint. So at the end of this trial, we wanted to see at the 21 days if there was still a difference in these two groups and if these differential profiles were sustained. So again, here we've got a comparison of the end of the liquid diet, time point A, compared to 21 days post-EEN in our unrestricted diet group. And again, we see good separation between these two groups, and the same comparison between CD treat and unrestricted group at day 21. So in a summary of our results, at the end of the liquid diet, all of these patients had not had any food for 8 weeks. They'd all been on enteral nutrition diet, and post-EEN or post-CD treat intervention, we have different metabolic profiles of our CD treat group and our unrestricted group. So this shows that we know the unrestricted group confers an inflammatory metabolic profile. So the fact that these two are different is promising and can be potentially used for looking into mechanistic how this works, but also looking at prognostic and potential early diagnosis biomarkers. So we can conclude from this study that Crohn's disease patients present a significantly altered fecal metabolome after 3 weeks following a normal diet. So take-home messages from both of these studies of celiac disease and inflammatory bowel disease. So we show a pipeline of candidate biomarkers for gastrointestinal disease using LCMS as a platform to discover. Clinical metabolomics is a useful tool for better understanding celiac disease and Crohn's disease mechanisms, and optimization and application of metabolomics approaches to the study of gastrointestinal disease provides a valuable insight into these interactions. So from the celiac disease study, we show that metabolic alterations were observed between celiac disease and healthy controls, with a panel of disease-specific metabolites highlighted, and also in response to a gluten-free diet. In inflammatory bowel disease, we show that we can look at potential biomarkers for evaluating response to dietary treatment, and that Crohn's disease patients present a significantly different fecal metabolome after 3 weeks compared to following a normal diet post-EEN.
Just shift it a little bit, just shift it, sorry. Sorry, now we're coming to the end of things. So in summary, we where we are at Strathclyde is we've got a range of trials that have gone on, and we're getting there with developing discovery-based biomarkers. As I mentioned at the start, these processes take a long time. They take years to develop the cohorts, to develop the tools to analyze those cohorts, and then to actually extrapolate the data and make sense of it all as well. And I think, tools like I said for the future directions piece there, you can see tools like AI, machine learning. We've kind of used those tools quite a lot in our area of science, even before they became really big buzzwords. Machine learning, PLSD has been around for good and for bad within the metabolomics community for 10, 15 years at least, probably longer. And I think we're going to constantly use those tools to develop the biomarkers within our cohorts. I think I'm a big advocate, and I think the group's a big advocate of that multi-omics approach, of being able to try and gather in as much biomolecular information as possible. There is no point siloing metabolomics away by itself to develop biomarkers. You really need to understand that full comprehensive change in chemistry that happens when an individual gets a disease, because it's only by understanding that and the profiles that we can really get to the bottom of what's going on. And that then leads forward to, as I said, personalized healthcare, where even with glucose, and using that as an example, you've probably seen, and probably some of you have got one of those glucose sensors that can ultimately feed into your mobile phone and track the glucose levels in your mobile phone. That technology is only going to become more expansive. The stuff that we discover, the metabolites that we discover, and the biomarkers we discover are only going to be translated onto those more daily, hourly, minutely updated measurements of how our health is going, because that is where precision and personalized healthcare is moving. So as a final takeaway, clinical biomarkers, they're transforming healthcare, and they have done for hundreds, if not thousands of years. But it's only really since we've got those tools and those analytical and digital technologies to be able to really, really understand things at scale and at speed as well. And it's only really by implementing them across the whole central dogma that we can really develop the biomarkers of the future that can hopefully, ultimately, help identify disease early and save multiple. And the last thing, a point to note at the moment before I open this up to questions back to Domenica and the team, is this is a really good paper down here. It's not from my group at all, but it gives a good summary of recent discovery level biomarkers in disease. If you're interested in that area, please have a look. At that point there, I think we're finished and we're open to, I tell a lie, we should really thank everybody first before I finish it off. So these are all the collaborators that we've got for the Fraley research that we've done. These are all the collaborators that we've got from the IBD research that we've done as well. And thank you very much to Dom and the EMN team, and also thank you all for listening.
Thank you very much, Nick and Tricia. That was a great and amazing talk from both of you. And I think we are now open to the questions. I really would like to encourage everyone to just put their questions in the Q&A box chat. And I think we can now go with the first question. So I think this is for Trish. So for the altered metabolites in gluten-free diet, what are the values you choose for fold change and p-value cutoff to be considered significant?
So for the fold change cutoff, we chose a fold change greater than 2, and for the significance level, just p less than 0.05. And we also had a criteria of using the VIP levels, so the variable projection from the OPL-SDA model of greater than 2 as well. So we had those 3 criteria to determine if a biomarker or if a metabolite was considered significant.
Thank you.
All right, and the next question is for the celiac disease study. Can you comment on why you used an OPL-SDA model over a PLSDA or an SP-LSDA? Also, how did you normalize, transform, and/or scale the data?
Yeah, so we used an OPL-SDA for all the studies that we use for our fecal samples and for our human studies. And we just use these as they take into consider all variables, and they're more reflective for humans, and especially when you're using different groups. So we chose an OPL-SDA for the celiac study as well as the IBD study. And what was the second question again?
For scaling.
How did you normalize, transform, and/or scale the data?
Yes, so for the for both studies, they were done in the same way. So the platform that we've used to do our data analysis just now is TableAnalyst. So we do our scaling and normalization in these. We use specific settings that we use for it, but we do normalize and transform the data.
I think to add a little bit more from the OPLSDA perspective, I mean, ultimately all OPLSDA does is it's similar to PLSDA, but it just finds the best angle, the best orthogonal angle to your data to get a little bit of separation. We don't use the sparse PLSDA methodology, I suppose ultimately because we want to pull in as much data as is practically possible. And I know for the sparse methodology, it's about being as parsimonious as possible with the model. It does have a place, definitely, and I think it's something that we use in different studies, but maybe just not in this one.
All right, so then next question is, in case study of COD, did you control the diet of the participants?
So for the EMN study, we didn't control the diet, but what we did was for the cohort that were on a gluten-free diet, we used GIP as a measure for gluten consumption. So our main goal here was to look at the effect of gluten, and so we thereby the exclusion of gluten from the diet. So in the cohort of treated CHCs patients, we ensured that gluten wasn't, we excluded all patients that were requested to follow a gluten-free diet but may not have. GIP is it's a super sensitive marker for gluten, and it stays in the body for a while. So any sign of GIP, we excluded those patients. But in terms of diet, we didn't look at any of the any other dietary components. It is super important and it affects a lot of things, but the primary goal for looking at the effect of the gluten-free diet was primarily whether gluten or not had been consumed. I know that food diaries had been taken for some patients, but for the metabolomics analysis, that's something that we've not confounded for just now, but it is a super important variable, and in future we are going to be incorporating more of the dietary research and looking at other ways that we can enhance our understanding and look at diet a bit more and look at different components and include that a bit further. But in this study, we've looked at whether or not gluten has been consumed, but that's only dietary component.
It's such a big part of all of these diseases, diet, though, isn't it? And I think as a group, so we're actually part of a bigger research group called the Bingo Group, which incorporates like 40 or 50. Yeah. It's like 40 or 50 researchers, clinicians, example patients, all sorts of individuals that can ultimately do that whole multi-omics, multi-clinical assessment of it. And I think one of the things that we are focusing on is diet, and it's the fact that not just gluten, for celiac disease, but for IBD disease, there's a whole range of other things that are in diet that have never been considered before. And I think it is a big focus that's not just us, but the whole community are turning themselves towards.
Awesome. And the next question is for Patricia. At what level have these metabolites been identified as per the MSI, and how did you perform the identification?
Yep, so currently they're at MSI level 2. So for the untargeted stuff, we have ran them, and we're actually in the process just now of developing our own in-house library so we can enhance that MSI level further. So now they are at MSI level 2, so we've ran them through Compound Discover, so software that is thermo-specific that we run our data through. So they've been matched to databases, Cloud, and a couple of other different databases for an MS2 match. So we've got Attention Time, MRZ, and other database matches for MSI level 2 just now. But we are currently working to develop an in-house library to give that identification level.
And Domenica's helped build that library just before she left as well, didn't you, Dom?
Yeah. Just as a general question from my side to Nick, because nobody has asked anything.
Thanks,
Oh no, wait, sorry, because I think we have another question. Sorry. It's from Christoph. So it's saying, so right now, most of your work seems to be explorative. Did you perform any functional test of the identified metabolites, for example, what they do upon gut organoids?
Yeah, so currently you're right in saying that it's all exploratory work that we've done so far. So we have still work to do on validating the metabolites that we've identified. So one step that we're doing that is to confirm that the metabolites that we're seeing are those metabolites. So by running these on the standard library and then by doing more functional characterization work. So yeah, we can do that in a lot of different ways. We've got some CACO2 cells that we're growing just now that we can do some testing on to look at functional properties. But it's future work that we'd like to get going, but just now they're initial exploratory metabolites that are potential candidate biomarkers of disease.
I think just to add to that as well, I think the bingo group, as we're going to call them, that we're part of, they're very keen to, cell modeling's fine, but I think they're very keen to avoid in vivo modelling, simply because we've got access to such good patient cohorts. So I think any work that we will get put into cell models, but will potentially then just get recapitulated back into the next clinical trial that comes along. So we're in that lucky stage where we don't necessarily have to do that animal modelling because we've got a continual pipeline of clinical trials that are going on in this area. One of the professors we work with he's way more successful than I am, and he's really, really good at getting funding to fund these clinical trials. So there's several more that are bolted on, and we're constantly reevaluating the features that we've detected and about how we're going to measure them, then go forward, and how we're going to implement that knowledge we've got in a previous trial into the next one.
And we have also, so the metabolites that we found that are potentially implicated in disease, just from a general literature search, we can see that they are important in intestinal functioning and have done different searches on how they might be implicated. So we have general ideas, we just haven't confirmed that in studies ourselves. So that's what we're
Great. There's one more question. First, the person said, hi, both, great work. Do you think your PLSDA models would tell a different story when cross-validated? How reliable are the Q2 values for prediction?
Yeah, so that I would say is one of the limitations in especially the celiac study. So the IPEN study for IBD, the Q squared values and model performance characteristics were looking quite good. The celiac C study, that was the biggest limitation, but it might not necessarily be a limitation. It might just be that is the results of the study, that the fecal metabolome isn't significantly altered between the two groups. And from the microbiome paper that we saw, that was also their conclusions, was that looking at the overall profile as a whole, it's difficult to tell the difference between these groups. So I think the lower of these values is potentially reflective of the fact that you might not be able to, as an overall look, tell the difference between these two groups. And that just shows the requirement of the specific biomarkers. So what we need to do is recreate these models just using the significantly altered metabolites, therefore making a model using just these significant ones, because the overall profile as a whole, the model characteristics, especially for the celiac, were slightly lower.
Thank you, Trish. So just as a last question from my side that I wanted to ask to Nick. So Nick, you mentioned multiple times and stressed the point on the importance of not just focusing on metabolomics, but also integrating metabolomics with proteomics. And I totally agree with that, actually. But I was wondering, also for the world community, would you recommend any specific tool for that you have found also based on your experience that is particularly helpful in this regard?
How long have we got to answer this question, Dom? You're opening a can of worms with that one. I think my view on how to fuse omics data sets together, I think there's two big broad schools of thought. I think you can fuse it together at the statistical level. And if you speak to groups like Jessica Laxey-Sue, they've got amazing new methodologies for looking at sort of metabolite and protein proxies, and there's a whole slew of different statistics that they do with their work to be able to do that sort of work. There's, on the other side of that, though, you can bring things together at the biological level. So you can have your individual streams of data that go all the way to that sort of understanding the chemistry, understanding the canonical pathways, and then do the crossover and do the map at that point. From a software perspective, I mean, a lot of the time, I'm going to go back to the sort of Neolithic and pre sort of civilization comment about the fact observation is your best friend with these things. is getting used to the literature and knowing the area a bit like Tricia has with IBD, beginning to know the area really, really well, and know the papers and know the pathways and know what is inferred by that disease, and seeing how the results you've got from both your data sets reflecting on that. I think there's no real substitute for that. There are statistical methods, absolutely. There's a range of them out there that can map data together. And that's absolutely fine, and I'm not an advocate against or for those, but I think there's no substitute for observing your data, looking at the data, understanding how that holds in the biological context, and writing your discussion around that, writing your paper discussion, your grant discussion.
All right, thank you so much again. That was really a great work and super interesting talk from both of you. And so, yeah, once again, thank you. And I would like to thank you also all the participants to this webinar. Again, this was the last one before the summer break. We will continue again, we will start again in September. And we will see all of you hopefully at the conference this year in Osaka.
Come and see Tricia's poster. Come and see Tricia's poster. So if you want to know more, come and see Tricia's poster.
Sure. So yeah, thank you, everyone. And I want to wish you a very nice continuation of your day. See you.
Thanks, everybody. Bye-bye.
Bye-bye. Thank you.
Bye.